Hello, dear participants. A very good evening to all of you. Hope you all are staying at your home and enjoying also. Is it visible and am I audible?
Yes, ma'am.
It's my immense pleasure to extend a cheerful welcome to you all. Your presence always makes us very happy. Today is the day we begin to learn to look through the eyes of others to find out and experience what the world is like from them. You know what's great about this event? It is very appreciative that the participants are liking the workshop of Consortium Biotech Workshop. How we are knowing this? We are knowing on the basis of your feedback and the interactions during the sessions. A very, very big thanks from all of us. Biotech family, mostly cordially welcoming you all. Therefore, now the time to undertake the fourth workshop under Consortium Biotech Workshop. Everybody knows this Consortium Biotech Workshop is a series of 5 workshops. 3 we have already completed, or 3 we have already discussed the various facts, such as workshop number 1, how to start the lab work under aseptic conditions, which we already dealt on 1st and 2nd of June 2020. Under workshop 2, how the microbes are fascinating, various aspects we have dealt with on 7th and 8th of June. How to study the beauty of nucleic acids, DNA and RNA both. This was already discussed on 14th and 15th of June. We all are assembled here for the 4th workshop, that is how the gene is expressed in the form of protein. This workshop is of 2 days, 21st and 22nd of June 2020. As we know very well that we are living in the era of biotechnology. Why we have designed all these workshops, as we discussed earlier also, because we all are doing some stupid mistakes whenever we are designing any project or any experiment. We are trying to just overcome such mistakes recently or presently in the form of digital mode, and if anybody is interested to do that in the form of hands-on, we will just provide them the standardized protocol also, and they will do by their own, that is hands-on, in the lab of the School of Biotechnology and Autonomous University Teaching Department, Rajiv Gandhi Pradyogiki Vishwavidyalaya, the University of Technology of Madhya Pradesh, Bhopal, which is accredited with grade A by NIC. Now, how the gene is expressed in the form of protein. This workshop has been divided into 4 sessions, followed by the feedback session. First day, that is today, we are discussing on the 2 sessions, that is cloning of DNA fragments, that is the session 1. It is having 3 types of clonings: molecular cloning, reproductive cloning, and therapeutic cloning. Myself and Riddhi Tiwari is going to share this session. Second session is mainly shared by Dr. Mrityunjay Singh and Dr. Suresh Kumar Jatwa on the basis of cloning vectors and expression vectors. Tomorrow, we will begin our session with screening of the colonies, that is blue and white screening by Aditi Parekh, and purification of proteins, followed by protein estimation, that is Shivangi Shrivastava and Yusra Siddiqui respectively. After the completion of all the 4 sections, then the session of feedbacks, whatever we discussed, whether that is really useful for you and which type of interactions you are doing during the presentations, we will just go through that. Why gene cloning? No 2 leaves of a plant or a tree is having the same genetic makeup. Although they are having the same environmental conditions, they are present on the same plant, same tree of the same branch, having same environmental conditions or atmosphere as well. Hence, in the middle of the 19th century, Gregor Mendel formulated a set of rules to explain the inheritance of biological characteristics. The basic assumption of these rules is that each heritable property of an organism is controlled by a factor known as a gene, that is, physical particle present somewhere in the cell. The rediscovery of Mendel's laws in 1990 marks the birth of genetics, aimed at understanding what these genes are and exactly how they work. For the first 30 years of its life, this new science grew at an astonishing rate. The idea that genes resides on the chromosome was proposed by W. Shatton in 1908 and received experimental backing from T.H. Morgan in 1910. T.H. Morgan and his colleagues then developed the techniques for gene mapping, and by 1922 had produced a comprehensive analysis of the relative positions of over 2000 genes on the four chromosomes of the fruit fly, that is Drosophila melanogaster. Then in the year 1971 to 1973, a whole new methodology was developed, enabling previously impossible experiments to be planned and carried out, if not with ease, then at least with the success. These methods referred to as recombinant DNA technology or rDNA technology or genetic engineering, and having at their core the process of gene cloning, sparked another great age of genetics. Such technology spawned over biotechnology, which put genes to work in the production of proteins and other compounds needed in agriculture, medicine, archaeogenetics, forensic science, and industrial production, etc. etc. But during the 1980s, when the excitement was at the peak, by the gene cloning revolution was at its height, it hardly seemed possible that another equally novel and equally revolutionary process was just around the corner. According to DNA fraternity, Kerry Mullis invented the polymerase chain reaction, that is PCR, during the drive along the coast of California one evening in 1985. Whenever such discoveries was done, then immediately clicks all these facts in our mind. What exactly does it mean to clone? Whenever we are planning to clone anything, how to clone, in which manner we have to proceed. There are three important things which are very, very essential to clone any gene. Number one, that is the gene of our interest, that is fragment of DNA, and in which we are going to clone, that is the suitable vehicle or the vector. Because there is a ratio of insert is to vector. What is the size of the insert and what is the size of the vector? On the basis of which we have to select. First, we have to select the size of that is our desired gene, and only on the basis of the size of that desired gene, what is the suitable vector. Then a ligation takes place between these two and the recombinant DNA molecule is formed. The third important thing which we have to select, that is the body of the host, that is host selection, which we have selected in this case is bacterium. Once the desired fragment of gene or the DNA is ligated in the body of the vector and form the recombinant DNA molecule that is ready to incorporate in the body of the suitable host which we have selected for our experiment. Once it is incorporated or transported into the host body, that is the process of transformation takes place. Now, this bacterium is carrying the recombinant DNA molecule. And further, multiplication of recombinant DNA molecule takes place inside the body of bacterium, and this bacterium is undergoing the process of cell division. Once this host is undergoing the process of cell division, that recombinant DNA molecule is also undergoing the process of multiplication after numerous cell divisions, resulting in a clone. That is in the form of that is agar-agar plating in the form of bacterial colonies. And screening of our desired one will be done on the basis of blue-white screening. This is the basis of genetic engineering, recombinant DNA technology, gene cloning, molecular cloning, DNA cloning. I'm repeating once again. First, we have selected the fragment of a DNA that is of our desired one. On the basis of the size of that DNA, the vector is selected. That is selection of the vector. Once selection of the desired gene or the fragment of the DNA and the selection of the vector takes place, then the process of recombination with the help of ligase enzyme, this desired gene is ligated in the suitable vector and then incorporated in the body of the suitable host. Once the bacterium is carrying the recombinant DNA molecule and the transportation of this recombinant DNA molecule into the host body, another process is multiplication of recombinant DNA molecule inside the host body. As the cell divides, the host cell is also dividing. Along with this multiplication of recombinant DNA molecules takes place. After numerous cell divisions resulting in a clone, that is in the form of bacterial colonies on agar plate. Simultaneously, as we just discussed about, the polymerase chain reaction was also evolved simultaneously once the cloning was evolved. Polymerase chain reaction, that is PCR. We know this PCR is based on three important steps. First, step one is denaturation at the highest temperature, that is 94 degrees centigrade for 1 minute. But I may mention here that DNA naturation, primer annealing, elongation of DNA polymerase, and whatever the program which we are setting in the PCR, these all depends upon the nature of the DNA, the size of the DNA which we have collected from which source. Right? We are just depicting this as a typical form of DNA, that is PCR, that is polymerase chain reaction. The step one is denaturation, that is for 94 degrees centigrade for 1 minute. Both the strands of this DNA, double stranded DNA molecule, has been denatured into the single stranded. Then step two is the primer annealing. From the 94 degrees centigrade, the temperature is lowering down to 56 degrees centigrade for 1.5 minutes. Annealing of the primers at the 3' end. Once the annealing of the primers at the 3' end at the lowering temperature of 56 degrees, then increasing of the temperature to 72 degrees centigrade for 1 minute. That is with the help of DNA polymerase and DNTPs. Elongation of DNA polymerase takes place, and both the denatured strands of the DNA, they have synthesized their complementary strands with the help of annealed primers. And as per the programs which we have set in the PCR, number of billions and trillion copies of such product was obtained. Again, question arises, why gene cloning and PCR are so important? Because we are looking for the cloning. So we have two methods, or we have the two ways we can say, gene isolation by cloning or gene isolation by PCR. That's why, although this gene cloning and PCR, these two are the straightforward procedure, but they are moving hands in hand. Why gene cloning and why PCR is so important? Because gene cloning, which we are doing with the help of, or gene isolation, which we are doing with the help of cloning, hence we have the suitable clone on the basis of the size of our desired vectors, on our desired insert or the desired gene or the DNA of interest. And in the same manner which we have just discussed, we have just formed the recombinant DNA, DNAs or recombinant DNA, that is plasmid, recombinant plasmid. Then this recombinant plasmid is transformed in the body of the bacterium for their multiplication. And as we know, screening takes place on the basis of the colonies which are growing on agar agar. That is white and blue screening, blue and white colony screening. Once we are doing this, there is one problem, or before that, we must know cloning allows individual fragments of DNA to be purified and is the only way of isolating long genes or those of never been studied before. This is the beauty of this experiment. What is the beauty? Two important things. Number one, cloning allows individual fragments of DNA to be purified and is the only way of isolating long genes or those of never been studied again, never been studied before. But there is one problem. What is that problem? The problem is how we can select or identify just one gene. Here we are having n number of fragments, but which one is our desired one? How can we know? With the help of this technique. For that purpose, we have to go for gene isolation by PCR. This is the fragment of the DNA having n number of genes, and this red one is our desired one. PCR allows production of a single gene or fragment in several billion times. After the completion of desired cycles, 20, 25, 30, whatever is the program which is applicable for that DNA, which we have set in the PCR machine, on the basis of which we are getting the several million times of that copies like this. This is our desired gene. With the help of polymerase gene reaction, this is the copies of our desired gene. But still, there are two important problems. Although sub-problems have been solved, but again two problems are there. What are they? Number one, PCR cannot be used to isolate genes that have not been studied before. PCR cannot be used to isolate genes that have not been studied before. But this problem was overcome by this method, because cloning allows individual fragments of DNA to be purified and is the only way of isolating long genes or those of never been studied before. And second problem, the PCR is not suitable for manipulating long genes. This problem is also overcome by the isolation, that is gene isolation by cloning. It means that's why these two methods, gene cloning and PCR, although they are having the standard procedures, but they are moving hand in hand. One is having some drawback, another one is overcoming that drawbacks. Another one is having some drawbacks, one is overcoming that drawback. So, gene isolation by cloning techniques is still in use. Just the glimpses of PCR, everybody knows what is PCR. First, extraction of DNA from any living organism, and as per the standard protocols, either we are extracting the DNA from microbes, viruses, plants, animal, human, then the extraction, isolation, purification, quantification, all have been completed as per the standard protocols. Then now we are having the PCR tube in which DNA sample, primers, nucleotides, Taq polymerases, and mixture buffer is present. All these we have kept in the thermal cycler, which is known as PCR, and program is set, suppose for 20 cycles or 30 cycles. After the completion of these cycles, we got the amplicons, that is amplified product of that DNA, which we are running on the gel, that is agarose gel. On the basis of the size of that DNA, the agarose gel is decided whether it is of 0.8%, 1%, 1.5, 2%, etc., etc. The bands which we are visualizing, we have to compare with the ladder, that is molecular weight ladder. These are bands of the sample. Why then is gene cloning still used? Because gene cloning is having their own characteristics or properties, and PCR is also having their own specific properties. Apart from that, to synthesize a primer with a predetermined sequence. That is, we can do that with the help of gene cloning. That is, with the help of PCR. Easy to synthesize a primer with a predetermined sequence, but if the sequences of the annealing sites are unknown, then the appropriate primers cannot be made. But in this case, PCR cannot be used to isolate genes, and that has to be done by cloning. As we discussed, unknown, we have to go with cloning. Known, we have to go with PCR. Limit the length of DNA sequence that can be copied by PCR. That is, 5 kilobases can be copied fairly easily, and segments to 40 kilobases can be dealt with using specialized techniques. But this is shorter than the lengths of many genes, especially those of humans and other vertebrates. For the longer gene, we know very well, cloning must be used for the longer gene. PCR is not suitable, cloning is suitable for the longer gene. Even if the sequence of a gene is not known, it may still be possible to determine the appropriate sequences for a pair of primers based on what is known about the sequence of the equivalent gene in a different organism. That we can do with the help of cloning, not with the help of PCR. That's why there is a necessity of gene cloning as well as PCR for the isolation of desired gene. Whenever we have selected our desired gene, now the second step is selection of the suitable vehicle or the vector. Two important vectors are there. One is cloning vectors and another one is expression vectors. As its name suggests, cloning vectors to clone a desired gene, and expression vectors just for the expression of clones which we have obtained with the help of cloning vectors. Now, the insert is fixed, that is selected, the vector is selected. Then third step is purification of DNA from living cells. In our previous workshops, we have discussed the purification, quantification, extraction, isolation, purification and quantification of DNA in viruses, microbes, plant, animal and humans. Here we are we are describing how the purification of DNA from living cells. We have taken the example of bacteria. After completing all the cells, now all the steps of the gene cloning, a culture of bacteria is grown and then harvested. Now we are having the culture of bacteria in the broth condition, that is in the liquid condition. For the harvesting of bacteria, centrifugation takes place. Usually the harvesting takes place at 10,000 or 12,000 RPM in the centrifuge. Pallet of cells are formed. Now the cells are removed and broken to give whatever are the components which are present inside the cell that should be free. Or we can say that cell lysis takes place so that the genetic material which is present inside the cell, that is coming out. Once that genetic material is present inside this cell extract, now DNA is purified with the help of suitable method. This purified DNA is ready for its quantification. Quantification is done with the help of spectrophotometer on the basis of optical density method. That is at 600 nanometer. We are just observing the optical density of purified DNA of bacteria. Now, we have selected our gene of interest, selected our vector, now purified our DNA. Now, Another step is manipulation of purified DNA. Why we have to manipulate the purified DNA? That is the basis of the recombinant DNA technology, which is having the ability to manipulate DNA molecules in a test tube itself, or we can say that in an append of itself. Why we are doing manipulation? How we are doing manipulation? We are doing manipulation with the help of purified enzymes for just to study DNA replication and transcription. What are the various DNA manipulative enzymes? Depending on the type of reaction. We have 5 categories, 5 classes of DNA manipulating enzymes on the basis of their type of reaction. Number 1 is nucleases. These are enzymes that cut, shorten, or degrade nucleic acid molecules. Nucleases, as its name suggests, working on the nucleic acids. So they are just cutting, shorten, or degrade nucleic acid molecules. Ligases, that is join nucleic acid molecules together. Modifying enzymes, remove or add chemical groups. Polymerases, make copies of molecules. Topoisomerases, poly means many, make copies of molecules. Topoisomerases, we are just introducing or removing the supercoils from covalently closed circular DNA. These are the 5 classes of DNA manipulative enzymes with the help of which we are manipulating our desired fragment of gene. Nucleases are mainly categorized under 2 categories or 2 classes, exonucleases and endonucleases. Whenever we are talking about the exonucleases, exo, that is outer side, towards the end of the DNA fragment molecule, the DNA fragment or the desired DNA fragment. Remove nucleotides one at a time from the end of the DNA molecule. How? This is the typical double-stranded DNA template. These beaded ones are considered as the nucleotide. In between the 2 complementary strands, these are known as the hydrogen bond. And between the 2 nucleotides, the phosphodiester bond is present. Because we are talking about the exonucleases. Exo means both ends. Here, right, red arrow is showing. This is the place where the exonucleases showing their doing their cleavage, nicking of single nucleotide. Once it is nicked, another one is ready for nicking of single nucleotide on both the strands, that is double-stranded DNA. Again, once it is over, then it is ready to nick from this place. Likewise, one by one, they are nicking at the ends of the double-stranded DNA molecule. Some examples of exonucleases are BAL31. It is working on the double-stranded DNA. That is, one by one, they are nicking on both the strands. Likewise, this one nucleotide is removed with the BAL31. Then that BAL31 is again digested with the DNA molecule. Then second nucleotide is removed from both the ends, 3' and 5'. Another example is exonuclease 3. This is mainly working on 3' ends of the double-stranded DNA. They are removing a single nucleotide only at the 3' end. It is not working on the 5' end. But lambda exonuclease, it removes nucleotide only at the 5' end of the double-stranded DNA molecule. And always remember, restriction sites, they are always having the palindromic sequences. What are palindromic sequences? These are similar sequences. I'll tell you in the later slides also. In the case of endonucleases, as we know, nucleases are of 2 types, exonucleases and endonucleases. As their name suggests, endo, that is inside, within the nucleus. The red arrows depicting the sites within the nucleus where the endonucleases works, and they are just dividing into or fragmentation of that DNA into the small pieces or the desired ones. That's why this endonucleases or restriction enzymes are considered as molecular seizures. Some examples of this endonucleases, these are S1 nuclease, it is working on single DNA, that is single-stranded DNA molecule as well as double-stranded DNA molecule. S1 nucleases, if it is digesting with, restriction digestion takes place with this S1 nuclease. At this point, what will happen? The nicking or the cleavage takes place at that particular point. Likewise, in the double-stranded also, one after the other, both the strands are nicking with the help of S1 nuclease. It means S1 nuclease is acting on single-stranded DNA molecule as well as the double-stranded DNA molecule. DNA is one, another example. It is also working on the single-stranded DNA as well as the double-stranded DNA. Single-stranded DNA, these are the places where the DNA is one is working in the single-stranded as well as in the double-stranded. And restriction endonucleases, that is same at the particular restriction site. They are showing the nicking at the particular site in the double-stranded DNA. These enzymes are found in the bacteria and archaea. What is the mechanism of cleavage of restriction enzymes? Restriction enzymes. These are double-stranded DNAs. Suppose we have taken the example of EcoR1. These are the places. This is the place of EcoR1, AATT and AATT. These are considered as the palindromic sequences. Same sequences. AATT on the mother strand and AATT on the complementary strand. This is the site where the EcoR1 in between A and G. Here also in between A and G, the nicking of the EcoR1 takes place. Once the cleavage of the EcoR1 takes place at this site, means restriction enzymes works on both the sites at a time. Just to nick both the sides of that restriction site. These are the restriction sites where this restriction enzyme works. When this cleavage of this takes place in this double-stranded DNA with the help of restriction enzyme EcoR1, such type of ends are developed, which are known as sticky ends. And they are, whenever nicking takes place, it means they are just to cut DNA. All restriction enzymes make two incisors, once through each sugar phosphate bonds are broken down in each DNA molecule. That is double helix DNA. Sugar phosphate bonds are broken down. Then only such type of nicking takes place with the help of restriction enzyme at the restriction site. All restriction sites are having a specificity and they are only located at the particular position, not everywhere, anywhere in the vector molecule, at a particular site, at the particular position. Why the name? Restriction enzymes. Because these, as their name, restriction, because this enzymes of bacterium restricts the replication of viral genome, therefore called as restriction enzymes. There is no entry of that viral genome which replicates with the genome of the bacteria. Totally restricted. That's why it is known as restriction enzymes. No restriction of viral genome inside the bacterium. That's why it is known as restriction enzymes. On the basis of structure, cleavage site, specificity, and cofactors. Four important categories, four important parameters, four important factors, that is structure, cleavage site, specificity, and cofactors. Types of restriction enzymes are classified. What are the types of restriction enzymes? Type 1, type 2, type 3, and type 4. Type 1 restriction enzyme, the cleavage site is random, around 1000 base pair away from recognition site. Location of amylase, that is endonucleases, and methylase located on a single protein molecule. Some examples of type 1 are EcoK1, EcoA1, CFRA1. These are some examples of type 1. Likewise, the type 2 is specific within the recognition site. That's why it is much and much in use as compared to other restriction types, because all our others are random in nature. Type 2 restriction enzyme is specific within the recognition site. Endonucleases and methylase are separate entities. Some examples are EcoR1, BAMH1, HIN3, commonly used restriction enzymes. Again, type 3 restriction enzymes, random in nature, 24 to 26 base pair away from recognition site. Similarly, with the first type 1 restriction enzyme, endonucleases and methylase located on a single protein molecule. Some examples of type 3 restriction enzymes: EcoP1, NIF3, EcoP151. Type 4 restriction enzyme cleave only methylated DNA and show weak sequence specificity. Their sites are still not very much well defined. Methylated, hydroxymethylated, and glucosyl hydroxymethylated DNA cleavage takes place 30 base pair away from one of the sites. Example is HAE4. After various types of restriction enzymes, the another class of enzymes are, that is, ligases. Very important. This is considered as seal the nicks that remains in the DNA molecule by forming a phosphodiester bond. It is also known as molecular glue. Suppose here the discontinuity appears in the DNA molecule, and this discontinuity is repaired by the restriction digestion, that is with the help of DNA ligase. Once the restriction digestion takes place with the DNA ligase, then this discontinuity is converted into continuity. That's why it is known as molecular glue. Suppose we want to join two molecules of DNA that can be joined or ligated with the help of DNA ligase. Hence, these two fragments of DNA are ligated with the help of DNA ligase. Polymerases, another class of manipulative enzymes. Polymerases synthesize a new strand of DNA complementary to an existing DNA or RNA template. Polymerization of nucleotides during synthesis. 4 categories: DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, reverse transcriptase. DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, reverse transcriptase. Basic reaction. Here we have the DNA template along with the primer at the 3' end. Primer is annealing at the 3' end, and newly synthesized strands takes place. These all red nucleotides are newly synthesized. How it happens? That is with the help of, the first example is DNA polymerase 1. There is a nick in between these two fragments of the DNA in the mother strand. We have to just fill this nick. What we are doing? We are applying the DNA polymerase 1. We are just doing the restriction digestion of this DNA template with the help of DNA polymerase 1. And this nick is filled along with whatever are the old nucleotides which are present here. Existing nucleotides are replaced with the newly synthesized nucleotides. These all are newly synthesized. It means DNA polymerase 1 is replacing the existing nucleotides with the new nucleotides and filling the complete gap or producing or synthesizing the new strand. Likewise, the no fragment, if we are applying this just to fill the gap, what it is doing, it is only working on the filling of this nick or filling of this gap. That is, CATT is only synthesized in between these two fragments of the DNA fragment of the mother strand, and it is not replacing the existing ones. only filling the gap. That is, fill the gap with the help of no fragment. Reverse transcriptase, that is applicable on RNA template. This is the RNA template. Again, primer is annealing at 3' end, and with the help of this primer, new DNA strand is synthesized, this which is complementary to this RNA template. And this DNA is, once this template is formed, this RNA template is removed, and this newly synthesized DNA is forming its complementary DNA template, hence known as complementary DNA. Apart from DNA manipulative enzymes, we have DNA modifying enzymes also. These DNA modifying enzymes are working only on the basis of either by addition or removal of specific chemical groups. Because these chemical groups are present at the end of the DNA template or the double-stranded DNA molecule or the single-stranded DNA molecule. Hence, they are known as end modification enzymes. DNA modifying enzymes, that is working on the addition or removal of specific chemical groups, which is present at the end of double-stranded or single-stranded DNA molecules. The first example is alkaline phosphatase. It mainly removes the phosphate groups at the 5' terminus of DNA molecule. And this phosphate group is replaced with the help of hydroxyl group. This is possible only with the presence of when the restriction digestion takes place with alkaline phosphate. The second example is polynucleotide kinase. This polynucleotide kinase having addition of phosphate groups at the 5' terminus. Here, the addition of 5 phosphate groups at the 5' terminus by the removal of hydroxyl groups. And terminal deoxynucleotidal transferase, that is, it adds or more nucleotides, it adds one or more nucleotides to the 3' end of the terminus of a single or double-stranded DNA molecule. This they are adding one or two, here one, here two, they are adding one by one the two nucleotides. This is here also 1, 2, 3. They are forming the tailing of nucleotides at the 5' at the 3' ends. And topoisomerases, as we know, introduce or remove supercoils from the covalently closed circular DNA. Once the nicking takes place in the DNA duplex with the help of restriction enzyme. For instance, we have selected the restriction enzyme, that is EcoR1. This EcoR1 cuts at this place, ETAA. Here, ETAA. That is, these sequences are known as palindromic sequences, which is present at the restriction sites, always present at the restriction sites. Once the nicking takes place at the restriction site, that restriction site is of EcoR1. Such type of unequal fragments are formed, which are considered as protruding ends, hence known as sticky ends. Here are the examples of sticky ends and the blunt-ended DNA fragments with the help of restriction enzymes by the process of restriction digestion. Hint 3, with the help of hint 3 digestion, we are getting the 5 protruding ends. With the help of PSTL digestion, getting the 3' protruding ends. And with the help of EcoR5 digestion, that is, blunt ends are produced. Now, sometimes what happens, our vector is having the sticky end or blunt end, and our insert is having either the sticky end or blunt end. It means the vector, suppose vector is having sticky end and the insert is having blunt end. One is having, that is, vector is having sticky end and the vector is, that is, insert is having sticky end. What will happen? It is not possible to ligate these two. Either one is having sticky, another one is having blunt. It means there must be same ends are present on their sides, on their ends for the process of ligation, right? For this purpose, we have three mainly components that are known as linkers, adapters, and homopolymer tailing. Linkers are chemically synthesized double-stranded DNA oligonucleotides having one or more restriction sites are already present on that short oligonucleotide sequences. Some examples of linkers are EcoR1, HIN3, BAMH1. These linkers are chemically synthesized double-stranded DNA oligonucleotides having one or more restriction sites already available on that linkers. Both the vectors and DNA are treated with restriction enzymes to develop sticky ends. It is not one is having the sticky end or one is having the blunt end, and we are just applying the linkers on the sticky end, then blunt end is going to attach. No, both the vector and DNA are treated with the restriction enzyme to develop sticky ends. Both are having the sticky ends, then only the process of ligation takes place. Both are having the same type of ends, that is sticky ends, then only the process of ligation takes place. The sticky ends are ligated with T4 ligase with very high efficiency to the termini of the vector to produce recombinant plasmid DNA molecule. This is the mechanism of linker. Typical linker, this is the BAMH1 site which is already present in the linker. Now, the use of this linker, here we have the blunt-ended molecule. We are using such linker which is already having BAMH1 site. Now, we have to develop that site also in this DNA template, that is double-stranded DNA molecule. Now, we are ligating these linkers with DNA ligase enzyme. These linkers are ligated at the ends of this double-stranded DNA molecule. Then the treatment, then restriction digestion takes place with the help of BAMH1. You just look at this. Sticky ends are produced with the help of this BAMH1 because this restriction site is already available in this linker. Hence, that restriction, when we are digesting with this restriction enzyme, that is already also available on the ends of this double-stranded DNA. Now, this DNA fragment is ready to incorporate or ligate in the suitable cloning vector. Adapters are like the linkers. They are also short double-stranded oligonucleotides carrying an internal restriction enzyme sites and single-stranded trails at one or both ends. This is the typical adapter, but there is one difference between the adapter and linker. This adapter is already having the sticky end, one protruding tail, another one is normal one. That is with the help of BAMH1 sticky end. One sticky end is already available in the adapter. This is not available in the case of linkers. This protruding sequences can be ligated to the DNA fragments containing a complementary single-stranded terminus. After ligation, then what will happen? DNA can be cleaved with appropriate restriction enzyme to create new protruding terminus. We are ligating this with the desired fragment of the DNA. Then what they are developing? They are developing, the DNA can be cleaved with the appropriate restriction enzyme to create new protruding terminus. Here is the typical structure of an adapter. This. Then we are just ligating with adapters. Adapters are in the same manner, the linkers are ligated on the ends of the DNA fragment. Then with the help of DNA ligase, these are just ligated at the ends, and with the help of polynucleotide kinases, then what will happen? This blunt ended are converted into the sticky end by the ligation of adapters. Now, again, this fragment is ready to incorporate in the suitable vector for further processing. The third type of molecule that is known as homopolymer tailing. Homo means same. Suppose if we are having the poly-deoxycytosine, poly-DC tails, these are attached to the vector, and poly-DG to the DNA to be cloned. Just look at this. Poly-DC is present here and poly-DG is present in the gene of interest. Both are ligated. Then what will happen? Both are complementary to each other, and recombinant DNA molecule having G and C at the respective sites produced. Once we have just selected our gene of interest, suitable vector, the DNA, recombinant DNA is ready, that is purified with the help of various manipulative enzymes and modifying enzymes, whatever is suitable, we have just applied on it and prepared our DNA. Now our DNA is ready to introduce into the living cell, that is in the body of the host. What are the sources of DNA which we are using to incorporate or to into the body of the living cell? The sources of DNA are genomic DNA, amplified DNA, synthetic DNA. Genomic DNA, which we have extracted from the cells and purified complementary cDNA by reverse transcription of messenger RNA. And amplified DNA using polymerase chain reaction. And synthetic DNA, that is DNA made artificially using a machine. Whatever we have discussed in our previous slides, this is the vector, this is the foreign DNA, both are of the suitable nature. Then insertion or ligation takes place and recombinant DNA plasmid is formed. Then uptake of this recombinant DNA plasmid by E. coli. This is the host chromosome. Now this vector is present inside the body of bacterium, that is the host. Then growth of this E. coli cells takes place in the liquid medium. Many copies of foreign DNA is present in this medium, and expression of foreign DNA in the presence of a suitable marker. Because whenever such type of colonies are formed, we have to focus on the blue and white screening colonies or white and blue screening colonies. Beta galactosidase reaction takes place. This beta gall acts on X gall, a clear soluble substrate, to produce a blue precipitate. We have to focus on the white because our desired gene might be in the white colonies, not in the blue colonies. Hence, there are two methods of the transformation of foreign DNA into the living cells, which is already incorporated in the suitable vector. Chemical transformation, electroporation. Excuse me. Chemical transformation, that is chemically competent cells are ready to incubate with the recombinant vector molecule, which is having the gene of our interest. Then, with the help of heat shock treatment, that is only for a few seconds. Suppose we are giving only for 4 minutes, 3 minutes, 10 minutes, depends upon the requirement of the experiment. Then what will happen? They are mainly breaking the cell membrane of the bacterium for the insertion of all these recombinant plasmids. Once recombinant plasmids are transformed inside the body of the bacterium, then inside the body of bacterium, they starts multiplication. Along with the multiplication, then they are ready for the recovery of the product. Now, what is the product? That is the recombinant plasmid is the product. Here, they are ready to just collect the recover the desired product. And this desired product is screening on the bases of the colony, which are developed on the agar plate. Bases of, again, blue and white or white and blue colonies. Excuse me. The another method is electroporation. Electroporation, here we have the competent cells which are ready for receiving the process of electroporation, that is electric shock. Again, the recombinant are ready to just incorporate inside the bacterium cell through electric shock. That electric shock is only for a fraction of seconds. Then what will happen? That recombinants are incorporated or transformed inside the body of that bacterium. Then that bacterium is undergoing the process of multiplication and ready. We are then that is ready to recover the product. We are just after the incubation, we are recovering our desired product which contains our desired fragment, that is the desired gene. Now, how to obtain a clone of a specific gene? But whenever we are doing all this, the biggest problem is problem of selection. This is the fragment of DNA which is having n number of genes along with our gene of interest. Nicking takes place with the help of EcoR1. Many DNA fragments are produced. These are inserted into the cloning vector. These all are having their various fragments of DNA along with our desired one. All will produce colony. How can we say, how can we isolate, or how can we identify our desired one? What? Just to overcome this problem or to solve this problem, two methods are there, or two basic strategies. One is direct selection, another one is library screening. Direct selection, that is only on the basis of recombinant which we want to survive, others not. And library selection, that is only on the basis of correct clone or correct clone library. That is with the help of clone identification. Direct selection, library screening on the basis of clone identification. Direct selection, here again we have the plasmid. For instance, we have P65 plasmid having the n number. These are the EcoR restriction sites. Restriction digestion with the help of EcoR1, 13 different fragments are formed, and these 13 different fragments are ligated in different vectors along with our desired one. Because this method is known as plating out method. Only the correct recombinant can survive, others will not. The simplest example which we have selected here is these simplest example of direct selection occurs when the desired gene specifies resistance to an antibiotic, that is chemomycin. Why others are not showing any appearance in the form of colonies? Because they are not suitable to grow on this medium which contains chemomycin. Only the gene of our interest which is having such type of colony, because in their vector they are having the chemomycin gene that shows its expression at the time of when we are just growing our colonies and screening the colonies on the medium which contains chemomycin. The another method is identification of the clone from a gene library. We are doing this for n number of purposes. Number one, analysis of the individual clones to identify the correct one. Problems of marker rescue. Another one is many bacterial mutants are not auxotrophs, and foreign genes from animals and plants sometimes do not function in the bacterial cell. For this purpose, there is a need of screening process to identify one particular clone containing the gene of interest from among the very large number of others in the gene library by using nucleic acid probe to screen the library based on hybridization with nucleic acids and analyze the protein product. Plague hybridization is the most suitable technique for the isolation of clones from a gene library. Search genomic library with a probe to identify clone obtaining desired gene. This is the filter. We are keeping this filter on the plaques of this colony. Now, this ideal probe labeled nucleic acid with sequence matching the gene of interest. This filter is keeping on the plaques, and this DNA on filter corresponding to plaques. This block filter with non-specific DNA or protein and hybridized to labeled probe detected by autoradiography. This is labeled with our nucleic acid, and that is having the positive hybridization among all these. This should be detected on autoradiography. For the genomic library, random digestion of genome by restriction enzymes, genomic DNA present in our desired sample of the living organisms, isolation of DNA from cells, these are the plasmid vectors, digestion with restriction enzymes, these are the cleaved vectors, these are the cleaved DNA, which we have to just ligate inside this cleaved vector. Ligation with DNA ligase, and these all recombinant DNAs are recombinant vectors are inserted into the bacterium. Amplification of genomic library, the genomic DNA library takes place, and that DNA is isolated in the form of in the separate test tubes or the separate ependophs in the form of gene library, which is having the fragments of various various lengths. Not all are having the same because random uh, digestion of genome by restriction enzyme takes place. But apart from all these advantages, we are having two major disadvantages of this method. One is non-coding regions, that is introns of DNA is also there. And second one is needle in a haystack. Various applications of gene cloning and DNA analysis, broadly divided in two categories, in research and in biotechnology. Under the research, mainly the applications of gene cloning and DNA analysis that are identifying the gene in a genome sequence, determining the function of an unknown gene, to study the transcriptome and proteome, the transcriptome, which is the messenger RNA content of a cell and which reflects the overall pattern of gene expression in that cell. The proteome, which is the protein content of a cell and which reflects its biochemical capability. Studying protein-protein interactions as well. Hence, we have the three broad categories in which under which we are studying or we are having the applications of research: studying gene location and structure, studying gene expression and function, studying genome. These are various applications of PCR, broadly categorized in three categories: molecular identification, sequencing, and genetic engineering. Molecular identification, that is for molecular archaeology, molecular epidemiology, molecular ecology, DNA fingerprinting, classification of organisms, genotyping, prenatal diagnosis, mutation screening, drug discovery, genetic matching, detection of pathogens. Sequencing, bioinformatics, genomic cloning, human genome project, and genetic engineering, that is site-directed mutagenesis, gene expression studies. The one most common technique which we are using for the just obtaining the expression of DNA, which we have obtained on the agarose gel. This was discovered by Edwin Miller Southern in 1975, and by its name, the technique is known as Southern blotting. That is transfer of DNA bands which are visualizing on the agarose gel on the nitrocellulose membrane. Extract and purify DNA from cell which you have selected. DNA is restricted with enzymes, separated by electrophoresis, denature DNA, transfer to nitrocellulose membrane, that is nitrocellulose blotting the paper, add labeled probe for hybridization to take place, wash off unbound ones, and radio, that is autography, just to visualize which one is the exact one of our desired one. DNA, which we have applied on the gel by the process of electrophoresis, which is the DNA is migrating from negatively charged to positively charged direction. Then this is the way in which we are just transferring our DNA band onto the nitrocellulose membrane. First, we have the trough containing the transfer buffer along with the platform, sarin wrap, Watchman filter paper. Gel is plated on the Watchman filter paper. On the gel, nitrocellulose membrane we are putting of the same size. Again, Watchman filter paper. Then the stack of the filter papers, and there is at the top, this black one is the weight of the appropriate size, so that that band can easily be transferred on the nitrocellulose membrane. Then when it is hybridized with the unique nucleic acid probe, then it is filtered and sealed in meal bag. Once it is filtered and sealed in meal bag, then what will happen? Remove unbound probe, and it is detected exposed on the X-ray film to filter, and that is detected on a autoradiogram. This is the molecular weight marker, and this is our gene of interest, which is easily transferred on the nitrocellulose membrane. This is known as southern blot analysis. Likewise, analog of southern blotting, northern blotting. This was discovered by two scientists, James Alvin and George Stark at Stanford University in 1979. There, in the same manner, the manner in which we did the southern blotting, we are doing the northern blotting, that is transfer of RNA on the nitrocellulose membrane. Like whatever we did, in the same manner, we are transferring the RNA bands on the nitrocellulose membrane with the help of same procedure. Then suppose we want to study the genome of either the whole genome sequences we have to study or the partial genome sequences we have to study. We can study with the help of Sanger sequencing or pyrosequencing. Applications of gene cloning and DNA analysis and biotechnology. 6 different categories of applications: purification of proteins from cloned genes, chemical industry, archaeology, agriculture, forensic science, medicine. Agriculture is considered as the world's oldest biotechnology. For we are using the gene cloning, DNA manipulation, genetic engineering, recombinant DNA technology, or molecular cloning for the following reasons. Number one, to improve varieties of the crop plant, for better nutritional qualities, higher yields. Gene cloning gives new dimensions to crop breeding, enable direct changes to be made in a genotype of a plant. These are some examples. Suppose plants are in a mood to make their own insecticides with the help of gene cloning. BT cotton is a very common example. Whatever the infection is available naturally, that can be overcome with the help of gene cloning or genetic engineering experiments. Genetically modified organisms, transgenic crops are there. If we want to improve our crops, we can do that is also with the help of gene cloning. Suppose we want to develop some specific characters, we also develop that specific characters with the help of gene cloning or genetic engineering and many more. This is the tomato, which is engineered with the help of CRISPR technology. CRISPR is the tool which has usually nowadays it is just flourishing with the colors for the gene manipulation, particularly the RNA manipulation or editing. In the case of medical biotechnology, production of recombinant pharmaceuticals, recombinant insulin, human growth hormones, recombinant factor 8, recombinant vaccines. Identification of genes responsible for human disease. These are tissue engineering for replacement organs, gene therapy for inherited diseases and other diseases as well, development of novel therapeutic molecules. This is also useful for diagnostics of various diseases and drug delivery system. In forensic science, the applications of gene cloning or molecular cloning are as follows: identification of crime suspects, then kinship by DNA profiling, and sex determination by DNA analysis. Chemical industry, production of bulk chemicals and solvents, that is ethanol, acetic acid, butanol, and acetone, and synthesis of fine specialty chemicals, such as enzymes, amino acids, alkaloids, and antibiotics. In the category that is archaeology, archaeogenetics, using DNA to study human prehistory. DNA can also be used to study prehistoric human migrations. These all we have collected from this book that is known as Gene Cloning and DNA Analysis by T.A. Brown. According to me, it is the Bible, it is the Quran Sharif, it is the Ramayana, it is the Gita of DNA cloning or gene cloning. Very good book, easily and very beautifully expressed each and every concept. Hence, as citizens of the world, we cannot ignore scientific research, but regulate it. Either we control gene technology today, or technology will redesign us by tomorrow. Hence, the cloning is the better tomorrow. Thank you so much for patience hearing. Now, I am inviting Riddhi Tiwari to continue this session for the remaining two types of molecular cloning, that is reproductive cloning and therapeutic cloning. We have discussed each and every aspect of the molecular cloning along with its applications. Now the remaining two types, that is reproductive cloning and therapeutic cloning, along with their applications, she is throwing light on it. Riddhi Tiwari. She is doctoral fellow of the School of Biotechnology. She worked at UK for a period of 6 years as a scientific writer. Riddhi Tiwari.
Thank you so much, ma'am, for such a wonderful presentation on gene cloning. And as you very well mentioned, that there are three types of uh, cloning, that is molecular cloning or gene cloning, that you have very well dealt with. And the other remaining two types are the therapeutic cloning and reproductive cloning, with which I'll be sharing some information about that. So it is the part of the fourth workshop of the Consortium Biotech workshop, that is how the gene is expressed in the form of protein. And in this, we will know something about reproductive cloning and therapeutic cloning. So whenever a layman comes across the word cloning, so immediately he or she thinks about the first mammal to be cloned, that was Dolly, the sheep, and they consider that cloning means producing animals in the form of clones in a laboratory. But that is not the case. But this kind of technology is called reproductive cloning. This is not the case that always the cloning happens in the lab, but there are many instances in the nature where we can see cloning. Like, for example, in the case of plants, algae, fungi, and protozoa, most of them reproduce through asexual reproduction, by which sometimes they produce spores, as you can see, that develop into new individuals that are genetically identical to the parent organism. Then there is one method of reproduction in bacteria that is called fission. That is the binary fission, where the bacterial DNA is replicated and the original cell is divided into two identical cells. Likewise, natural cloning also occurs in animal organisms during processes such as budding, fragmentation, or parthenogenesis. The most common and the most visible ones to us is in the case of identical twins, which are formed in humans and other mammals. This is a type of natural cloning. The identical twins are produced when a fertilized egg splits, creating two or more embryos that carry almost identical DNA. These identical twins have the same genetic makeup as each other, but they are genetically different from either of their parents. So, what was the era before reproductive or therapeutic cloning? Before many years of experiments that led to the creation of Dolly, it was thought that normal animals could be produced only by fertilization of an egg with a sperm. These germ cells are the only cells in the body that have their genetic material all mixed up and in half the quantity of every other kind of cell. That means when the haploid cells come together at fertilization, they produce one cell with the full complement of DNA. When it is joined together, that cell is termed diploid for twice or double. So these two halves, n plus n, makes a 2n. That means a whole. All the cells in the body have the same genetic makeup. When the one cell embryo duplicates its genetic material, both cells of the now two cell embryo are genetically identical. When they duplicate their genetic material, each cell at the four cell stage is genetically identical, and therefore all the cells in a particular adult will have the same genetic makeup, whether the cell belongs to lung or bone or blood or any other organ whatsoever. So first of all, let us know what is reproductive cloning. Reproductive cloning is the formation of one or more animals or humans from a body cell of another animal or human. These clones are genetically identical to each other and also to the donor of the cell from which the clone is derived. So they are exactly like the identical twins. Because they are genetically identical, the most commonly used cells are from the skin, and that skin can be grafted between members of a clone without rejection. The first cloning of any animal happened in the year 1958 in the case of frogs, and the first mammal to be cloned was the sheep Dolly. It was produced in 1997 by Professor Ian Wilmot with his colleagues at Roslin University, Edinburgh, Scotland. Subsequently, many other mammalian species have been cloned. So let us have a look on how which type of animals have been cloned till date. These are some of the animals. Most many other animals have been cloned. So as I mentioned earlier, the first one was the frog. First mammal was the sheep. Thereafter, cow, goat, pigs, rhesus monkey, ox, wild goat, and mouse. Different types of animals have been produced in the lab with the help of reproductive cloning. And the reasons for the cloning of such animals are in the case of various applications, which we will see later. So let us have a glimpse about the important years, about the important history of the animal cloning or reproductive cloning. So as I mentioned, in 1952, it was the first when the frogs were cloned, attempt was made. Then in 1997, the announcement of sheep called Dolly was made. Then after Polly was cloned, and it had a genetically altered human gene. Then in 1998, 50 mice were cloned in 3 generations from a single mouse. So this was a great achievement that from single mouse, 3 different generations were gained, giving 50 mice. In 1999, 3 goats were cloned. They were genetically altered so as to make protein in their milk, which will help to treat heart attacks and strokes, etc. In 2000, pigs were cloned for their use in human organ transplantation. In 2001, attempt was made to clone human embryos, but they could not grow larger than 6 cells. In 2002, a company named Clonade said it had cloned the first human, but it seemed to be a hoax. Next, another in 2004, Korean team tried to clone human uh, first human embryo. So what is the procedure of reproductive cloning? Reproductive cloning is based on the method of SCNT. What is SCNT? It is somatic cell nuclear transfer. In this procedure, a single body cell is isolated and the nucleus of that cell is transferred by extremely small needle into an enucleated unfertilized egg. As a result, the nucleus of the body substitutes for the normal combination of egg and spermatozoon so that the egg cytoplasm contains a transplanted nucleus and can develop into just the same kind of individual as the donor nucleus was taken from. By taking the nuclei from several cells of the same individual, a clone of animals is produced and now they are all genetically identical. If an animal from which the donor cell is taken remains alive, it is entirely possible to have clonal progeny that will be identical to the donor individual but completely different age. It would be as if you have 2 identical twins differing in age by 40 or more years. This is the method by which any animal is cloned. The example taken here is from the dolly. The donor cells from the sheep's udder were procured, and an egg cell was procured from a female sheep. The egg cell was enucleated, that is, nucleus was removed from the cell. Now, these 2 cells are fused together with the help of electric shock. These fused cell then multiplies to give rise to an embryo. That embryo is implanted into the foster mother to give birth to a cloned animal. But we need to know some facts that most clone animals are abnormal. Why? Because when the donor nucleus is taken from an adult or specialized cell, the probability of obtaining a normal animal decreases dramatically. So you can say that practically no more than 1 or 2% of all the nuclear transfers carried out from the adult cells yield normal adult individuals. And the remaining 98% develop abnormally and die at various stages of development. So nearly all cloning experiments that start with adult cells give abnormal results. It is virtually possible that this would also be true if if experiments were carried out on humans. Therefore, it would be unethical to carry out human cloning experiments because all the resulting individuals would suffer from some kinds of abnormalities. And many of these would be so severe that the fetus would never be born. Kindly mute your microphone, or that early death would certainly arise. It is because of very high incidence of serious developmental abnormalities. It is thought that the cloning of humans to produce a complete individual is absolutely unacceptable and unethical. So, can humans be cloned anytime? So, from a technical perspective, cloning humans and other primates is more difficult than other mammals. What's the reason? The reason is the proteins essential for cell division, known as spindle proteins, are present very near to the chromosomes in the case of primate or human eggs. So, the removal of the X nucleus to make room for the donor nucleus also removes the spindle proteins that interferes or hampers the cell division. But in the case of other mammals, the two spindle proteins are spread throughout the egg. Therefore, the removal of the X nucleus does not result in the loss of spindle proteins and the cell division carries out normally. Similarly, some external factors such as dyes and the ultraviolet light used to remove the X nucleus have the possibility to damage the cell and prevent it from growing. Likewise, there are some medical issues also with the with reproductive cloning. Those are that the enucleated cell and the transferred nucleus might not be compatible at all. An egg with a newly transferred nucleus may not begin to divide or develop properly. Implantation of the embryo into the surrogate mother might fail, leading to the failure of the pregnancy. Similarly, the clone animals tend to be having a larger size at their birth as compared to the natural counterparts. So, such animals are known to be suffering from large offspring syndrome. So, the clones with LOS or large offspring syndrome have abnormally large organs, and these large organs can lead to breathing problems, blood flow, and other physiological and metabolic problems. In cloning, the transferred nucleus doesn't have the same program as a natural embryo. So, the program is determined by the scientist. So, so if there is some problem in the programming, then there might not be a normal or near normal development. We all know that normally when the cells divide, the chromosomes get shorter. But in the case of clone animals, the telomere lengths were found to be longer, and there were no clear answers for that. So we can say that the chromosomes from the cloned cattle or mice had shown to be having longer telomeres than normal ones, and that might be a reason of early aging. So another aspect is of human cloning. So human cloning still appears to be a part of science fiction movie. Why? Because in 1998, scientists in South Korea, they claimed that they have cloned a human embryo successfully. But when they were interrogated, they said that the experiment halted earlier when the clone was just a group of 4 cells. In the year 2002, a, a Canadian-based company called Clonaid, they announced the birth of a first clone human, a girl called Eve. And not only one, they had claimed to have cloned more than 12 human clones. But after repeated investigation by the research community and the media, they could not prove their claim. Similarly, in 2004, another group in the University of South Korea, they published in a paper of journal of high repute, that is Science, in which they claim to have created a clone human embryo, but again they were they failed to prove their claim, and subsequently the paper was retracted from the journal. So, what are the pros and cons of reproductive cloning? The pros or the advantages of reproductive cloning is that any defective gene, if present, that can be eliminated. If there is any traumatic injury, then it will help in the faster recovery. It can also help in the elimination or treatment of infertility. Likewise, the clone body parts can serve as a backup system for humans. Like, for example, if somebody knows that he or she is going to have a genetic disorder related to, say, a heart or a liver, so if they know beforehand, then they can just prepare a clone of their body part and reserve it and use it whenever required. Next is another advantage is it can definitely combat genetic diseases, and it is used and it can be further used in the field of research purposes and for the alterations of plants and animals for getting for the case of preparing therapeutics from them, or provide for getting some growth hormones or interferons and substances like that. And we can also produce people with desirable traits. But what are the disadvantages of reproductive cloning? As I mentioned earlier, because of longer telomere lengths, there is a possibility of early or faster aging. And then there is a reduced sense of individuality. If any person's clone is produced, then they would be completely similar to each other. And then the cloned individual would not have its individuality. So, in this case, it will reduce its sense of individuality, which will lead to the overall reduction of the value of human life. Most importantly, reproductive cloning can hamper ecological, social, economical diversity, and thereafter the ability of adaptation. Then, production of undesirable traits can be done by some notorious people, which will invite malpractices into society, and the other ethical and religious beliefs are against this reproductive cloning. They say that the in this case the humans will act as god. But there are some fruitful applications of reproductive cloning also. So let us know them one by one. The reproductive cloning allows farmers and ranchers to accelerate the reproduction of their most productive animals to prepare or to produce safe and healthy food. It will help to reproduce the healthiest animals, which will reduce the use of antibiotics, growth hormones, and other chemicals, by which the consumers will be able to procure the animal products such as meat and milk, with, which would be more healthier, consistent, and safe. For example, a sheep has been genetically modified to produce milk that contains human protein essential for blood clotting, and scientists hope that this protein could be purified from the milk and administered to the such humans whose blood does not clot properly. Another most important advantage of using cloned animals would be in the case of drug testing, because these clones will all be genetically identical, which means that whenever a drug is administered, the responses would be in a uniform fashion as compared to a variable one observed in the case of animals with different genetic makeups. One more important application is it can be used to protect endangered species, like which are going to extinct sooner or later. In 2001, researchers produced the first clone of an endangered species, a type of Asian ox known as boar, but sadly it died just after its birth. Next, in 2003, another endangered type of ox called benteg was successfully cloned. Thereafter, some African wildcats were cloned, and in China, they have stored and they have preserved the panda cells because they think that if the panda is at the verge of extinction, then they can use that cell and produce clones of panda. That was about reproductive cloning. Now, what is therapeutic cloning? Therapeutic cloning is an extension to reproductive cloning where we do not produce or prepare a whole animal, but we just prepare the stem cells. So, this is a very upcoming field and it suggests that the stem cells could save your life. So, what is therapeutic cloning? Therapeutic cloning involves the creation of clone embryo just to produce embryonic stem cells with the same DNA as in the case of donor cells. These stem cells can be used in experiments to understand the disease and to develop new treatments. The richest source of embryonic stem cells is the tissue formed during the first 5 days after the egg has started to divide. At this stage, the embryo is called the blastocyst. There, the stem cells can be harvested and result in the destruction of the embryo while it is in the test tube. Production of cells by nuclear transplantation would be helpful for the replacement of body cells which no longer function normally. An important aspect is that an individual could be provided with cells of their own genetic makeup, but they would be in a more rejuvenated and healthier condition. So, the procedure is quite similar to reproductive cloning. Here also, uh, egg is removed from a donor female and DNA is procured from the skin cells and the enucleated ovum is fused with the DNA of the skin cells with the help of electric shock and it stimulates the cell division. This is the pre-embryo which would divide for near about 5 to 14 days and the stem cells can be extracted. The stem cells are then encouraged to grow into first they are conditioned in such a way to grow into specific organs and then which are transferred to the patients. Till date, scientists have been able to differentiate stem cells into almost all of the 220 cell types in the human body. So, as I said, somatic cell is taken from the patient, enucleated oocyte is obtained from the donor, it is fused together with the help of electric shock, embryonic stem cells develop, and they can be conditioned to be into different types of cells like pancreatic islet cells, hematopoietic cells, cardiomyocytes, neurons, and hepatocytes, and those cells can then be administered to an immunologically compatible patient through the transplantation. So this is basically human therapeutic cloning. But we should first know that how the stem cell differentiate. Stem cells or the embryonic stem cells are pluripotent in nature that they can differentiate themselves into any kind of cell. The control is basically by the growth factors, cytokines, and epigenetic processes such as DNA methylation and chromatin remodeling. The division of the embryonic stem cells happens in two types. First, one is a duplicate stem cell in which the process of self renewal occurs, and then the other daughter cell which will differentiate. The daughter cells divides, and after each division, it becomes specialized. After it reaches a mature condition, like if it becomes a red blood cell, then it will not further divide. See the in the figure also you can see that this blastocyst is derived from the embryo. Then this is the process of self renewal, and the other daughter cells have the property of pluripotency and they can differentiate themselves into three germ layers like ectoderm, mesoderm, and endoderm. Ectoderm will result into the formation of brain cells, spinal cord, nerve cells, hair, skin, teeth, sensory cells of eyes, ears, nose, mouth, and pigment cells. Mesoderm will give rise to muscles, blood, blood vessels, connective tissue, and heart. Endoderm will give rise to the gut, that is pancreas, stomach, liver, and other organs like lungs, bladder, and germ cells. What is the use of these embryonic stem cells? Embryonic stem cells can be used to grow healthy tissues in the laboratory to replace injured or diseased tissues. Therapeutic cloning allows for the production of specific organs that are almost 100% compatible with the patient in need of an organ transplant. Whenever organ transplant happens, then the patient needs to be administered with some immunosuppressive drugs so that he or she does not reject the transplant. But the side effect would be cause of hypertension, dyslipidemia, hyperglycemia, peptic ulcers, liver, and kidney injury. But the medication will not always guarantee organ acceptance. So, in this scenario, therapeutic cloning will play an important role by eliminating the possibility of organ rejection as it eliminates the need of dangerous immunosuppressant drugs. Additionally, individuals donating organs would not have to be inconvenienced by pain or a shortened lifespan in order to donate the organ. Therapeutic cloning can also be used to treat a wide range of diseases and disorders such as diabetes and Parkinson's disease. Similarly, invasive surgery would be can be avoided if a therapeutic cloning is successful because it will be able to repair damaged organs. The molecular causes of disease can be discovered by studying embryonic stem cells from the clone embryos of organisms with a particular disease. So these are some of the diseases which can be cured with the help of therapeutic cloning. I have gathered the important ones like Parkinson's disease, diabetes, muscular dystrophy, paralysis, Alzheimer's disease, heart disease, and genetic disorders. But sometimes the therapeutic cloning is not viable also because the adult cells are limiting. So, therapeutic cloning will lie on the extraction of embryos. And of that extraction, just a small portion of stem cells are usable for cloning. Likewise, some cells can mutate themselves and cause tumors. In order to cure any disease or treat any disease, millions and millions of eggs are needed. But unfortunately, we are currently do not have this type of supply of eggs. Again, the ethical issue of the religious sentiments arises that it would be against God's wishes. Important technical aspect is the extraction of eggs because it would the extraction of eggs from the female is quite troublesome and it's costly. Therefore, the cost of therapeutic cloning is very high. So, the new research which is going on nowadays is based on the replacement of the embryonic stem cells. So, the question arises that can the embryonic stem cells be replaced? So, to overcome the ethical and immunological challenges associated with human embryonic stem cells, iPSCs, that is, induced pluripotent stem cells, have arisen in the form of a promising alternative. So, this technology, the pioneer of this technology was Dr. Yamanaka. In 2006, he introduced about the 4 specific genes called MYC, OCT34, SOX2, and KLF4 that would encode transcription factors that could convert an adult mouse somatic cell into pluripotent stem cells. And these factors are named after his name and they are called Yamanaka factors. Uh, he was awarded the 2012 Nobel Prize along with Sir John Gordon for the discovery that the mature cells can be reprogrammed to become pluripotent. The human induced pluripotent stem cells can be harvested from individual patients, and the immune rejection can be avoided because the stem cells will be procured from the patient himself and will be administered to himself or herself again. So they will be the self donors. So that can be a form of personalized medicine. There are various sources of such kind of pluripotent stem cells, such as umbilical cord blood cells, bone marrow cells, peripheral blood cells, fibroblasts, keratinocytes, and cells present in urine. They can be differentiated into tissue-specific cells of desired lineages. Like we can, you can see, we can procure the pluripotent stem cells from different types of cells like fibroblast, hepatocytes, urine sample, blood cells, etc. Then they will be programmed using special reprogramming factors like this. Now I mentioned about the Yamanaka factors and other small molecules. Then the suitable delivery technique will be adopted. It could be via retrovirus, episomal vectors, direct protein, or the synthesis of mRNA. Then the colony will be formed, and then the iPSCs would be characterized into two types. Either it will be a positive cell line or a negative cell line. The positive cell line then can be further used for different uses like drug discovery, disease modeling, and regenerative medicine. And the negative cell lines are those cell lines which could not be reprogrammed. So those are failures. So likewise, fibroblast is taken in this example. So we can use different factors like integrating viral transfection, example retrovirus or lentivirus, non-integrating viral transfection like Sendai virus, adenovirus, episomal virus, mRNA, cDNA, plasmids, self-excising vectors like Crelox or piggyback transposon, or non-integrating non-viral factors like chemicals like valproic acid, sodium butyrate, vitamin C, microRNA, piRNA, siRNA, CHLP, AZA, and protein delivery. So all these factors in combination or individually could be used for the reprogramming of pluripotent stem cells. So, in a nutshell, we can see that how fertilization takes place in normal development, like one end and one end, like the sperm cells and the oocyte cell, they fertilize together to produce a zygote, which gives rise to a blastocyst, and it gets implanted into the uterus of the animal, and an adult is formed. But in the case of reproductive cloning and therapeutic cloning, the adult cell is taken and the donated oocyte is enucleated, that is nucleus is removed. Then nuclear transfer happens in both the cases. Blastocyst is formed in both the cases. But in the case of reproductive cloning, it is transferred to the surrogate to give birth to a clone. While in the case of therapeutic cloning, the blastocyst will give rise to ESCs or embryonic stem cells, which would differentiate themselves into different tissues. So this is all about reproductive cloning and therapeutic cloning. And I would end my session by saying that we are not humans by our biological origins or past. It is not in our material details of our lives that we find humanity, but instead it is our interaction with others in a social sense. It is love and compassion. Thank you so much. And just to sum up this whole session of cloning, today Professor Dr. Archana Tiwari and myself dealt with the three types of cloning. She explained about the gene cloning, that we know that the molecules of DNA from one or different species will be inserted into host organisms to produce new genetic combinations that would be of use in science, medicine, agriculture, and industry. And later in the second part, we studied about the reproductive cloning and therapeutic cloning. Reproductive cloning, as I said, it produces a clone animal or an organism, and therapeutic cloning is the production of embryonic stem cells or pluripotent, induced pluripotent stem cells. Um, and now the session is open for any types of queries. And after the queries, I would like to invite Dr. Mrityunjay Singh, who is the assistant professor at School of Biotechnology, RGPV Bhopal. So he will carry on with the further session of cloning vectors. So thank you all of you for your patience.
Excellent presentation, Riddhi, and thanks for your kind words. Due to
Dr.
time constraints, we can take queries at the end of both sessions.
Okay.
Dr. Mrityunjay, please wait for a minute.
Okay, ma'am.
Some questions are there in the chat box. Kindly entertain that questions.
Okay, ma'am. I'll also go through, and I would request Dr. Mrityunjay also to look, and Dr. Suresh, if I leave some queries.
Sure.
Yeah, ma'am.
You are just satisfying through the writing the answers of that query,
Riddhi,
so that he is going to continue his session.
Riddhi,
Okay, ma'am. Yes, yes, Suresh.
there is a question that what are stem cells?
And I have explained everything about stem cells.
So please you explain once again. One of the participant wants to know what are stem cells and what is adult cell, and another query is endonucleated donor oocytes.
Okay, the stem cells that I already explained are the cells which are usually obtained from the embryos, and that are known as embryonic stem cells. And they have the pluripotent competency that they can differentiate into any kind of cells in which they are conditioned into. This is about the stem cells. And what was the other cell? What was the other question?
Sorry?
What was the other question? Adult cell.
Adult cell, adult.
Adult cell means the cell from any adult, like that can be a skin cell or a fibroblast or any nerve cell. That is the adult cell. The cell which is procured from an adult individual is an adult cell.
Okay. And another question is what is the difference between totipotent and pluripotent?
So totipotency is the mechanism when it can differentiate into any kind of cell, and pluripotency.
Can I address this question?
Yes, ma'am, please.
Totipotent. Toti means total.
Total, yeah.
All the cells of the body can be can be obtained from the totipotent, and we can develop an organism from the totipotent. There is only one place in the body at the time of embryonic development, that is the 3 to 5 days old embryo. That is the place from where we can collect the totipotent stem cells. Other reproductive phases, we are having the multipotent stem cells. Always remember, only one place, that is 3 to 5 days old embryo, that
Yeah.
is having the capacity to have the totipotent stem cells. As its name suggests, toti means total. We can produce all the cells of the body and develop into an organism from the totipotent stem cells. Thank you.
Thank you, ma'am.
Thank you,
Another
ma'am.
question is, how do we stimulate stem cell to differentiate into a particular organ? How do we stimulate stem cell
Yeah.
to
Yes.
differentiate into a particular organ?
This is made by the different stimulant, like the transcription factors and the other proteins and peptides that just now I told about. So this is the way they are stimulated. Thank you.
Another question is, please, ma'am, explain how electric shock fuses the cell.
That is the high voltage of current we are just passing for a fraction of seconds. Once we are passing the high voltage of current for a fraction of seconds, that immediately disrupts the plasma membrane for the transformation of recombinant plasmid.
Thank you, ma'am.
Thank you, ma'am.
And another question is the same like earlier answered, that this cell is used for totipotency.
That ma'am has already answered.
Already answered.
There is no cell. There is a developmental stage, 3 to 5 days
Stage.
old embryo. From there, we have to just take the inner cell mass, right?
Ma'am, another question is, what is the difference between expression vectors and screening transfection vectors?
What is the question?
What is the difference between expression vectors in screening transfection vectors?
Like, kindly correct the question. Screening is
Question
different.
is not so. Yes, yes, ma'am.
Transfection vectors are the cloning vectors through which we are doing the process of transfection. Am I right? Because transformation is a step which we are doing at the time of cloning, just to incorporate the foreign DNA into the suitable vector and then transforming in the body of the host cell. That is known as transformation.
Okay, ma'am. Ma'am, another question is, why BT cotton and BT brinjal bend?
Only because of such type of characters developed which are not suitable for the society. That's why the government has banned.
Okay, thank you, ma'am.
What else?
Another question is, what is LOS? Riddhi.
LOS is large organ syndrome, which is observed in the cloned animals. They have large organs as compared to the natural counterparts.
Somebody is asking about pluripotency. You told me the meaning of pluripotency. Can I please know about pluripotency? Totipotent, pluripotent, multipotent. These are the three broadly characterize the stem cells. Pluripotent cells, almost all the cells of the body, but not all. Multipotent, some cells of the body, but not all. And totipotent, all the cells of the body to become an organism. This is the basic difference between these three.
Ma'am,
Unipotency is uni means one. Only one type of stem cell. Unipotent, pluripotent, आपका multipotent and totipotent. Right? Unipotent is only one type of stem cells which we are using.
there is a question, ma'am, please explain therapeutic techniques to use paralysis.
The answer to this is that this will be with the help of the stem cells. Like the stem cells of it would be neuronal cells would be generated and then administered to the patient, and
May I?
then the
May I?
yes, ma'am, please.
Actually, whatever the mechanism Riddhi has explained wonderfully, that is, whatever the somatic cells of the body, whether they are fibroblasts, whether they are neuronal cells, any cells of the body, they are having the tendency when we are just undergoing the process of reprogramming of the cell. Am I right? That is with the help of four factors: OCT4, OX2, KLF4, and CMYC. These are the four important factors which are considered as epigenetic factors. These are having the tendency to convert any of the somatic cells of the body into induced pluripotent stem cells. Right? In the case of paralysis, if neuronal cells are in a position of defect in nature, then they can be treated with the help of induced, they can be converted into first into the induced pluripotent stem cells, and then can be treated with the help of that induced pluripotent stem cells. I think I made it clear. I think so. Am I right? Any other queries? Some queries just written by anybody, any participant.
Uh, what exactly the gene mapping and gene sequencing? What is the purpose?
Whenever we are having the complete genome of any organism, because all the genes are located on a map that is known as genetic or genomic map. Whatever the complete genes available in any organism, that all are located at a particular positions in the gene. That's why we are saying this gene is located at this position, from this nucleotide to this nucleotide. This is its sequence. Number of base pairs. Likewise, interons all are available in a set of genes, or in a proper manner. Exon, intron, exon, intron. We are having the complete genetic material, which is mainly useful for to study how many genes are present, what are the various functions of these genes, in which manner they are behaving. Because all the activities of our body is under the control of specific genes. N number of applications we have already. If we want to study the genome, if we want to study the genome analysis, that's a complete whole genome analysis we can do with the help of genomic map. Partial sequence analysis we can do with the help of genomic map. If we want to go for any disease, suppose that gene is having some defect which is causing a disease, we can do that also with the help of genetic engineering. That is sequencing, right? N number of applications are there related with the sequencing. The latest sequencing is pyrosequencing, 4 detector based, ATCG. All are having the separate detectors. Thank you so much. Anybody else? If anybody is having the query, you please write it down in the chat box so that we can start the second session. Otherwise, we are getting late. Am I right?
Ma'am, what is genetic polymorphism?
Genetic poly means many. That is genetic variability occurs in the morphism. That is genetic that is structure. That is known as genetic polymorphism. Poly means many. Variations occurs in the structure of any species, any organism, a set of species. Any other question?
No, ma'am.
Sure.
Uh,
Yeah, we are going
One
to
question.
start with this.
Yeah.
Yeah, one question is there, please.
Ma'am, just now I saw one question regarding Yamanaka factors. So these are nothing but the factors which have been discovered
It's
by the inventor, and these are basically four basic specific genes that code for the transcription factors, and these genes are MYC, OCT, SOX, and KLF4, and these act as the factors which will help in the reprogramming of the somatic cells to convert them into pluripotent stem cells.
induced pluripotent stem cells.
Yeah, induced pluripotent, exactly.
Any other?
Ma'am, I think we should start the next
Yeah,
session of Dr. Mrityunjay. Yeah.
yeah. Please.
Okay, thank you, ma'am. Good evening, all. Now we are discussing about cloning vectors. Then, why cloning vectors require? Cloning vector is used as a vehicle to artificially carry foreign genetic materials into another cell where it can be replicated and expressed. It is used to amplify a single molecule of DNA into many copies. Cloning vectors are DNA molecules that are used to transport clone sequences between biological host and the test tubes. Without cloning vector, molecular cloning is totally impossible in earlier days. The molecular analysis of DNA has been made possible by cloning of DNA. The two molecules that are required for cloning are DNA to be cloned, which is the insert or desired gene, and a cloning vector, where we can multiply the number of that gene or genetic material. A cloning vector is a small piece of DNA taken from a virus or a plasmid or the cell of higher organism that can be stably maintained in an organism and into which a foreign DNA fragments can be inserted for cloning purposes. Many vectors, most of the vectors are genetically engineered. The cloning vector is chosen according to the size and type of DNA to be cloned. Means which cloning vectors are using, we are going to use is depend upon the insert or desired fragment of DNA which are going to, which we want to multiply them. For further experiments or analysis, the vectors therefore contains features that allow for convenient insertions or removal of DNA fragment in and out of the vector. For example, by treating the vector and foreign DNA with a restriction enzymes and then ligate the fragment together. After a DNA fragment has been cloned into a cloning vector, it may be further subcloned into another vector designed for more specific uses. This picture shows the vector in gene cloning, where this is the vector or cloning vector, and this is the insert which is isolated from the cell, and using the same restriction enzymes which cut this plasmid, we digest the genetic material and collect that gene of interest. And by ligating this insert and vector, we form the chimeric or recombinant DNA plasmids. These plasmids here shows E. coli having two types of genetic material, bacterial chromosome and plasmids. From the plasmid isolate and cut by restriction enzyme, same restriction enzyme, and combine with desired gene of interest. And this recombinant plasmids is put plasmid into bacterium by transformation. The recombinant bacteriums allow bacteriums to reproduce and produce the clone of cells and that gene. History about this cloning vectors: scientist Herbert Boyer, Keiichi Takura, and Arthur Ricks, working in Boyer's lab in University of California, recognized a general cloning vector with unique restriction site for cloning in foreign DNA and the expression of antibiotic resistance gene for the selection of transformed bacteria. In 1977, they described the first vector designed for cloning purposes, that is PBR322, a plasmid vector. This vector was small, 4 kilo base pair in size, and had two antibiotic resistance genes for selection. Feature of a cloning vectors: all commonly used cloning vectors have some essential features, having origin of replications. This makes autonomous replication in vector. ORI is a specific sequence of nucleotide from where replications start. Due to this, the plasmid is automatic replicate inside the bacteria. When they are going to divide or grow, When foreign DNA is linked in the sequence along with vector replication, foreign, that is desirable DNA, which we want to clone, DNA also start replicating within host cell. Second is cloning site, that here shows multiple cloning sites. Cloning site is a place where the vector DNA can be digested. This is the vector. It digested with the help of different restriction enzymes. But when we are going to make a recombinant, we are using same restriction enzymes for both. It is here multiple cloning site means having so many sites for different restriction enzymes which are used to digest your gene of interest. It is point of entry or analysis for genetic engineering works. Recently, recombinant plasmid contains a multiple cloning site which having up to 20 restriction sites for restriction enzymes. Now, third one is selectable markers. Selectable markers is a gene that confers the resistance to particular antibiotics or selective agent that would normally kill the host cell or prevent the growth. Like here, antibiotic resistance gene, which are used by bacteria to grow in solution where the antibiotic is added in medium. Fourth one is reporter gene and marker gene. That reporter gene are used to cloning vectors to facilitate the screening of successful clones by using feature of these genes that allow successful clone to be easily identified. Such features present in cloning vectors is used in blue-white selection. Additional properties of vectors: it should be short, small, compatible with host cell, incompatible with other vectors, should become high in copy numbers because you require multiple number of your copies of your desired gene. It should able to express itself utilizing the host machinery by using origin of species, and it should be able to move under two systems in prokaryotic and eukaryotic systems. Now, type of cloning vectors. Here listed different types of cloning vectors. We are using plasmid, bacteriophage, plasmid, bacterial artificial chromosome, that BACS, yeast artificial chromosomes, that YAC, human artificial chromosome, that is HAC, or mammalian artificial chromosome, that called MAC, and retroviral vectors. Now, one by one, we discuss all plasmids. First one is plasmid is autonomously replicating circular double-stranded extra chromosomal DNA, which is physically separated from a chromosomal DNA and can't replicate independently. They are most commonly found in bacteria. Sometimes they are present in archaea and eukaryotic organisms. Most general plasmids may be used to clone DNA insert of up to 10 kilobase pair in size. Means by using plasmid, we can multiply our gene of interest, which having maximum 10 kilobase pair in size. Many plasmids have high copy numbers, and high copy number is useful as it produces greater yield of recombinant plasmid for subsequent manipulations. However, low copy number plasmids may be preferably used in certain circumstances. For example, when the protein from the clone gene is toxic to the if number of copy number is high, in these cases that kills the cell and they didn't grow. Examples of plasmids are PR322, PUC1819, F plasmid, whole plasmid, etc. Here, the pictures of PVR322 and PUC1819 are shown. PBR 322 is having 4361 base pair long sequence. Both are circular. PVC 1819 having single differences where 19 is different from 18 by only the restrictions, these multiple cloning site orientations is opposite in 19 in respect to 18. Size of representative plasmids, here the list of plasmids are written, and here size, nucleotide length in kilobase pair and molecular weight and organism. Here PVR 322 having 4362 base kilobase pair, 4362 base and 4.362 kilobase size, and its organism where it's present in E. coli. F plasmid is E. coli, tall in Pseudomonas butyrata, and here copy number of some plasmids. For F plasmids, copy number is low, only 1. F1 prophase having 1. RK2 is 4 to 7. The organism is E. coli. PVR 322 having 16 to 20. PUC 18 is copy number in between 30 to 50, and PIJ 101 is 40 to 300. Now the nomenclature of plasmid cloning vectors. The name PVR 322 confers with the standard rule for vector nomenclature. P indicates that this is indeed a plasmid. PR identified the laboratory in which the vector was originally constructed. DR stands for Bolivar and Rodriguez, the name of two researchers, the two researchers who developed PVR 322. 322 distinguishes this plasmid from other developed in same laboratory. There are also plasmid called PVR 325 and 327. PVR 322 contains two marker resistance gene, that ampicillin and tetracycline, and origin of replication along with different restrictions sites. Why plasmids are good cloning vectors? Because it's a small size, easy to manipulate and isolate, circular structures which are more stable, replication independent of host cell, because due to origin of it's not depend upon the chromosome of bacteria to divide. Several copies may be present, facilitate replications, frequently have antibiotic resistance, that is detection assay. Disadvantage using plasmids: cannot accept large fragments. Size range is from 0 to 10 kilobase pair. Standard method of transformations are insufficient. Now second is bacteriophage. The bacteriophage used for cloning are phase lambda and M13 phases. There is an upper limit on the amount of DNA that can be packed into phase, which is around 25 kilobase pair. Sometimes its maximum is 53 kilobase pair. There are also a lower size limit for DNA that can be packed into phase, and vector that is too small cannot be properly packed into the phases. This just represents the bacteriophage having head, collar, tail structures. Phase lambda is bacteriophage or phase, that is bacterial virus that uses E. coli as host to grow. The structure is that of a typical phase, head, tail, and tail fiber. Lambda viral genomes having 48.5 kilobase pair DNA with a 12 base single stranded DNA sticky end at both ends. Means the genome of this lambda virus having both end 5' protruding end of 12 base long, single stranded, that is sticky and complementary to each other. The both ends, these strands These genes are complementary and in sequence and can hybridize to each other to form cos sites or cohesive genes and form circular structure. Infections. Lambda tail fibers absorb to the cell surface of bacteria and receptors. The tail contract and the DNA is injected inside the bacteria. The DNA circularizes and lambda begins its life cycle in E. coli host. There are two kinds of lambda phase vectors, that insertion vector and replacement vectors. Insertion vector means contains a unique cleavage site whereby foreign DNA with size of 5 to 11 kilobase pair may be inserted, and in case of replacement vector, the cleavage site flank a region containing genes that is not essential for the lytic cycle of bacteria viruses may be deleted and replaced by DNA insert in the cloning process. Here, this region shows a replaceable region which is not essential for the lytic cycle and replaced by target or desired DNA. And after that, packaging and assembly take place. Now, M13 phase vectors. M13 vectors are used for obtaining single-stranded copies of cloned DNA, which are suited for DNA sequencing and in vitro mutagenesis. In this case, the gene of interest which are going to clone is single-stranded. And as we know, in the process of sequencing, we require single-stranded genetic material for sequencing purposes. They are derived from the 6.4 kilobase pair genome of the filamentous bacteriophage M13. Very large insert can be cloned in these types of vectors. Pure single-stranded copies of DNA of double-stranded inserts are obtained. Now, phasmid. Phasmid or phasmid is a plasmid that contains an F1 origin of replication from F1 phase. It can be used as a type of cloning vector in combination with filamentous phase M13. A phasmid can be replicated as a plasmid and also be packed as a single-stranded DNA in viral particles. This is the structures of phasmid that contains ampicillin resistance gene, double-stranded DNA origin, F1 origin, and these promoters secretion signals and code proteins and display peptides region. Now, phase vector present two advantages over plasmid vectors. They are more efficient than plasmids for cloning of large DNA fragments. Means is the difference from plasmids. We can clone the large fragment in respect to plasmid vectors. The largest clone insert in lambda phase is 24 kilobase, while the plasmid vectors is having less than 10 kilobase. It is easier to screen a large number of phase plates than bacterial colonies for identifications for recombinant vectors. Now, cosmids. Cosmids are plasmids that incorporate a segment of bacteriophage lambda DNA that has the cohesive ends that cos sites, which contains elements required for packaging DNA into lambda particles. It is normally used to clone large DNA fragments between 25 to 45 kilobase pair. They can replicate as plasmid if they have the suitable origin of replications. They can also be packed in phase capsids, which allow to foreign genes to be transferred into the cells by transduction by method of bacteriophage when infect the bacteria. This picture shows cosmids contain ampicillin resistance genes, polylinkers, phosphatides, and poly. Advantage over the plasmids: high transformational efficiencies, and cosmid vectors can carry up to 45 kilobase pair inserts. whereas plasmid and lambda phase vectors are limited to 25. Now, yeast artificial chromosomes, that is CI. The yeast artificial chromosome vector is capable of carrying a large DNA fragment up to 200 kilobase pair, but its transformation efficiency is very low. Cloning vehicles that propagate in eukaryotic cell host as a eukaryotic chromosomes. Final chimeric DNA is linear DNA molecules with telomeric ends, that is artificial chromosomes. Here, this is YAC vectors. By degrading the BAM-H1 and EcoR1 to this and target DNA by EcoR1, we find desired gene which we are going to insert, and this linear YAC vector and just ligate it. YAC cloning vehicles often have a bacterial origin of DNA replication, that is ORI, and selection markers for propagation of YAC through bacteria. Like selection markers are already ampicillin resistance gene is there. The YAC can use both yeast and bacteria as a host. What is the benefits of using YAC? Two, it can, first one is it can carry large size of insert, that is up to 200 kilobase pair, and it can use both yeast and bacteria as host. Now, bacterial artificial chromosome vectors are similar to standard E. coli plasmid vectors, contains the origin and gene encoding that ORI binding proteins required for plasmid replications, derived from naturally occurring large plasmids, that is F plasmids, low copy number, that only 1 to 2 copies per cell is present. The bacterial artificial chromosomes usually insert size is in between 150 to 350 kilobase pair. BAC refers for different kind of genetic studies of inherited or infectious disease because they accommodate much larger sequence without the risk of rearrangements and are therefore more stable than other type of cloning vectors. This is the diagram of BAC having size of 7 kilobase pair. Now, by how we use this, the bacterial F plasmid is cut within the lag Z gene with restriction enzymes, and the gene of interest is cut by using same restriction enzymes. Just combine or ligate these fragments, that BAC vectors and gene of insert. F plasmid and gene of interest ligate form recombinant BAC. BAC is electroporated into E. coli cells. Then cells are plated on X-Galia IPT medium, where white colonies are composed of transformation. They are transformed cells because it's LAGZ negative genes, because we cut the LAGZ gene by restriction enzymes and insert our desired gene. Then the original LAGZ protein, the coding product are the product of protein, or protein product of that LAGZ gene are hindered and not produced, so that it not form the their proteins, and that white colony shows LAGZ negative cells. Bacterial artificial chromosomes, where T1 derived artificial chromosomes, PAC is, which is called PAC. PAC was developed by Lano et al. In 1994. The constructed vectors incorporates features of both T1 and F- plasmids and can be transformed into E. coli by electroporation. In a PAC vector, insert of the size having 100 to 300 kilobase pair can be cloned. It is devoid of problems such as instability of clone DNA. The advantage of BAC compared to EAC, it is stable, easy to transformation, speed of growth of E. coli host, simpler to purify, more user friendly, and they are helpful in development of vaccines. Now, human artificial chromosomes. Human artificial chromosomes may be potentially useful as a gene transfer vector for gene delivery into human cells. It is tool for expression study and determine human chromosome functions. It can carry very large DNA fragment. There is no upper limit on size for practical purposes. Therefore, it does not have the problem of limited cloning capacity of other vectors. It also avoids possible insertional mutagenesis caused by integration into host chromosome by viral vectors. This is the pictures of human chromosomes contain centromeres, telomeres, replication origin, and gene of interest. Now, retroviral vectors. Retroviral vectors are used to introduce new or altered gene into the genome of human and animal cells. Retroviruses are RNA viruses. The viral RNA is converted into DNA by viral reverse transcriptase and then is efficiently integrated into the host genome. Any foreign or mutated host gene introduced into the retroviral genome will be integrated into the host chromosome and can reside there practically indefinitely. Retroviral vectors are widely used to study oncogenes and other human genes. The examples are lentiviruses, adenoviruses, herpes simplex viruses, and so on. What determines choice of vectors? Four factors are there: insert size, vector size, restriction size, or restriction sites, and cloning efficiency. For different type of vectors, the insert size is listed here. The application of different cloning vectors. Plasmid vectors having naturally occurring multi-copy plasmids. Size of insert is less than or equivalent to 10 kilobase pair. It is used in subcloning and downstream manipulations, cDNA cloning, and expression assays. Phases, that example is bacteriophage lambda. The insert size is in between range of 5 to 20 kilobase pair. It is used in genomic DNA cloning, cDNA cloning, and expression library. Plasmids, that plasmid contains a bacteriophage lambda phosphides. Replication size of insert is 35 to 45 kilobase pair. Genomic library constructions. Application is genomic library construction. Now back, bacterial artificial chromosomes. That is Escherichia coli F factor plasmids. Size of insert range is 75 to 300 kilobase. Analysis of large genomes. YAG is host cell is Saccharomyces cerevisiae, centromeres, telomeres, and autonomously replicating sequences. Range is 100 to 1000 kilobase pair, that is 1 megabyte. And it is used in analysis of large genomes. React transgenic, like example is React transgenic mice, and that is MAC or mammalian artificial chromosome or human artificial chromosome. Range of more than 100 kilobase pair, and it is under development for use in animal biotechnology. These are the references. Thank you all for patiently listening. Now, any questions, please write in chat box. We'll answer after the end of next session. Now I invite Dr. Suresh Kumar Jatwa for next presentation. He is assistant professor in School of Biotechnology, RGPB Bhopal, and discuss about expression vectors. Thank you.
Thank you very much, Dr. Mrityunjay, for a nice presentation. So, good evening, all. So, in this session, we will discuss about the expression vectors, their utility, their properties, and how we construct a expression vector, a good vector. This topic is a part of the fourth workshop, that is how the gene is expressed in the form of protein. So we will discuss about the expression vector, its property, like what is the difference between a cloning vector, as I discussed earlier. That's just previous session. That is the cloning vector. So what is the advancement, or what is the difference between a cloning vector and expression vector? So like the properties having promoters or spindle Garno sequence, or ribosomal binding site, transcription stop signals, origin of replication, polyadenylation, polylinkers, and selectable markers as well. So if we want to clone anything, so if we are having a vector, cloning vector, so this is simple cloning vector having some multiple cloning sites, means polylinkers. So here the sequence having various recognition sites for the different restriction endonuclear enzymes known as molecular seizure. So this is the gene of interest. This is gene of interest which you do want to insert or incorporate in this vector and clone it. So with the help of molecular season, we can open this frame by cutting with restriction endonuclear enzyme, a specific enzyme, and we can insert it, we can ligate it with the help of ligase, DNA ligase enzyme I discussed earlier session. So once you have ligated or inserted your gene of interest in the vector, now you want to, there is a need to transform in a host cell. So it may be a prokaryotic cell and it may be a eukaryotic cell. So after transformation, this culture takes place. After the culture harvesting, you can be able to take a product of clone or maybe gene of interest as a clones. So we can say overall the vectors are nothing. These are the vehicles for DNA insert and allows so DNA insert to be expressed efficiently. So if you see, this is a particular expression vector where you can see all the sequences for which makes it expressive, having this vector like operators, promoters, regulators, polylinkers, selectable markers, origin of replication site, ribosomal binding site, start and stop codon, all these things combined known as ORF, like open reading frame. So this type of vectors generally engineered to introduce a specific gene into a target cell. This can confiscate or may use the cell's mechanism for protein synthesis to produce a protein encoded by a targeted gene or inserted gene, which you have inserted here. So in addition to all these things, the gene of interest, these expression construct also contains regulatory elements, which you know, which you can say the open reading frame, which contains the start and stop codon, the operators, promoters, regulators, all these things. So these are the most efficiently needed for the transcription of gene of interest. So these may be plasmids or may be modified viruses. So you can use it for the expression. So expression vector must have the, if you see here, this is a simple vector. So you are, if you are constructing this particular vector of plasmid as the expression vector, So when the simplest expression vector construct, also known as transcription vector only. If this particular vector having the both properties like transcription and translation, means this the gene of interest can express in the form of protein, means after translation. So this particular vector known as the expression vector. For transcription and translation, a promoter and terminator sequence are must or needed, that has to be incorporated in the vector sequence. So these promoters of the expression vectors must have switch on, switch off mechanism, so we can control the expression of the entire gene or incited gene product, because the excessive amount of product of the gene of interest can also be dangerous or maybe toxic to the cell. How do expression vectors work? So it is having strong promoters, by which the promoters are responsible for the binding of RNA polymerase. So with the help of this particular sequence, the initiation of transcription can take place. So with the help of strong promoters, strong termination codons, or inserted transformation sequence, portable translation initiation sequence, or maybe adjustment of distance between the promoters and cloned genes. So all this type of vectors can be used. Expression vectors. So if you see this pictorial ray diagram, here it is. I would like to explain better way with the help of ray diagram. This is a vector in this form. So this is the regulatory gene, which having the control of the another gene for the regulation of another gene. So regulatory gene, promoters, this is RBS, ribosomal binding sites, where the ribosome can get binds. So this is the important, that is the coding sequence. So start codon or stop codon must be there. Terminator. So antibiotic resistance genes for the selection or the selectable marker, origin of replication. So in this way, if you are constructing a vector or expression vector, so you have to keep in mind all these type of property must be there in the vector. So once you have prepared a vector, constructed a vector, so this is your gene of interest, which you want to express in the form of protein. So once you have incorporated in the vector or maybe expression vector, this has to be transfected into the host cell. It may be prokaryotes like bacteria, or mammalian cell, or maybe yeast cell. So you are having different goals, different things in your mind while you are constructing a vector. So as per your requirement, like you want to first thing is with the vector, you wanted to incorporate or insert a gene of interest in the vector. So for this purpose, it is having MCS as discussed in earlier session also, that is the multiple cloning site. It has a various restriction sites for various restriction enzymes like molecular seizures. Once this insert plasmid into cell, enable the plasmid to replicate. So this has to be replicate inside the host cell and select for cell carrying the plasmid. So these are also the important thing. Very sorry. So for this purpose, you are having origin of replication in the vector and selection markers or screening markers like antibiotic resistance gene in the vector for this purpose. So after this, transcription of mRNA from the plasmid, if you are constructing expression vectors, that has to be expressed in the form of protein. So it has to be transcribed in mRNA. So for this purpose, the vector having promoters may be constitutive or indivisible, operator terminators. So with the help of this can be transcribed, because operators have the site for the RNA polymerase binding. After transcription, the translation of the mRNA into protein, it is also important step. So for this purpose, this particular vector must have RBS, that is ribosomal binding site, and start codon and stop codon. So promote proper folding nascent protein. So for this purpose, you are having co-expression of chaperones, solubilized in text, custom designed synthetic RBS, codon optimized ORF. ORF is nothing, it is open reading frame, which contains all these things like start, stop codon. For this detection or the purified target proteins, you are having like epitopes, tag histines or reporter GFP vectors. So this is the pictorial diagram of expression vector having all the respective properties which makes it expressive or expression vector. So first thing is the promoters. This vector must have promoter. That is the different thing with the cloning vector. So a promoter sequence always ensures a reliable transcription of the gene of interest because it has a sequence where RNA polymerase gets bind. Here you can see this is the promoter region. So the RNA polymerase can gets bind here and starts transcription in this direction. So the strong promoters are also necessary for an efficient mRNA synthesis with RNA polymerase. Here you can see the diagram, and the regulation of promoter is another critical aspect which should always be kept in mind while you are constructing a expression vector. So in E. coli, the promoter is regulated in two ways, like induction and repression. So promoters may be constitutive promoters and indivisible promoters. So a constitutive promoters is a kind of promoter which is unregulated, means it allows a continual transcription of its associated genes. It has not regulated, it is not controlled. While individual markers and promoters having a controlled or a regulated manner by which the expression of the gene associated with them can be controlled with the help of switch on or switch off facility of particular stage of development or a certain point of time. So these are some of the example of promoter like leg promoter. So this is leg gene. So leg Z gene is responsible for the production of beta galactosidase, and it is induced by the IPTG, that is isopropyl beta thiogalactosidopyranose. So this can be used, but a drawback of this particular is also there. So it is quite weak and cannot be utilized for the high level of production of the desired proteins. Like genes carry out the basal level of transcription even in the absence of inducers. So another example is the tryptophan promoters and these tech promoters. So another thing is that the vector must have polylinkers. This is also have been discussed in the earlier session. This is nothing, that is the MCS, that multiple cloning sites, which allows the restriction endonuclease enzymes to open the circle for the insertion of a targeted gene. Here you can see these are the structural genes responsible for protein alpha. This is for protein beta. This is for protein gamma. And these red ones are the ribosomal binding sites or ribosome binding sites. So with this ribosome binding sites follows the promoter, and it is responsible for efficient translation of the clone gene. The translation in eukaryotes is initiated at a particular sequence called Kozak sequence, and ribosomal machinery for this particular for the translation of mRNA is assembled at this site known as Kozak sequence. Signed Dalgarno sequence, here you can see this is the signed ribosome bind on the mRNA. This is binded to the mRNA. So here you can see the translation also initiate with the help of ribosomes. It needs some sequence, that is signed Dalgarno sequence. It is within the ribosome binding sequence only, known as signed Dalgarno sequence. So this conscious sequence of translation initiation site includes a set of 4 to 8 base pairs present upstream of the start codon. Poly A tail, it is also very important step while expression vectors, you are dealing with the expression vectors. So poly A tail present at the, you can see at the end of the mRNA here like this formed. It always protects mRNA from the degradation by the exonuclease enzyme or maybe endonuclease enzyme. It is extremely critical for stability of mRNA. It always needed. So it is also responsible for termination of transcription and translation. It allows the mRNA for the stabilization or mRNA production, and a nucleotic enzyme complex and a polymerase are prerequisite for the addition of poly A tail at the end of the mRNA like this. So here you can see like this, this is the sequence structural gene from mRNA, and after poly A tailing like this is translated into a specific protein. So reporter gene. So here you can see this is a regulatory sequence. This is including promoters, operators, all these things. So this is the reporter gene, which encode for a particular protein that has to be detected or quantified. So this is the reporter gene, and this is the tool to measure efficiency of the gene expression in the form of protein like this. While transcription is made mRNA, and after translation in the form of protein. So this amount of this reporter gene can easily be measured. So it is a tool, and detect the intercellular localization of the protein, the rate of expression, all these things can be identified with the help of reporter gene. So overall we can say that for the expression we are having two types of expression system, like prokaryotic and eukaryotic. So overall, even either prokaryotic or either eukaryotic, both have some advantages, some disadvantages, which can be always taken into the consideration while constructing an expression system. So there is no such expression system which can be considered universal for a heterologous protein expression here like this. So the prokaryotic expression system. So as all we know, E. coli is widely used prokaryotic expression system. It expresses high level of protein, and the strains of the E. coli could be manipulated genetically for as per your need for the production of recombinant proteins, so that they are rendered safe for large scale experiments and maybe fermentation. So purification of proteins has become easier with the help of this. And prokaryotic expression system, there are still many difficulties associated with challenges caused by the production of protein from the cloned foreign genes. It is due to some characters of the inserted gene or maybe some property of the host cell. And eukaryotic expression system, challenges posed when cloning gene in a prokaryotic expression system can be overcome by using eukaryotic expression system. So with the eukaryotic expression system, these includes yeast, mammalian cells, etc. So the advantages while using the eukaryotic expression system, maybe protein product of the cloned gene is expressed at high levels. Proteins can be easily purified by using particular tests which are included in the vector itself, like histamine, etc., etc. So disadvantage also having, if we are having some advantage, so disadvantage also there. So disadvantage is the eukaryotic cell grow at very slow pace as compared to the prokaryotic cell. So by using these vectors, there's some applications are there. If you want to study, or identify a particular protein localization like this, and protein purification, if you want to develop a recombinant protein or isolate or synthesize fusion proteins, or it has also some medical uses. These are some of the references. Thanks. So this is the end of the second session, and now I would like to invite Dr. Mrityunjay Singh, Assistant Professor, School of Biotechnology, RGPB Bhopal, for the sum up of the second session. Dr. Mrityunjay, please.
Thank you, sir, for discussing all about expression vectors. Now, in this session, we discuss about cloning vector and expression vectors. As we already discussed different types of cloning vectors, that plasmid, M13 phase, lambda phase, cosmid, phase mid, back, AI. And in case of expression vectors, it's having tightly regulated promoters, that is like UV promoters, to stimulate transcriptions, that artificial inducers, that IPTG is added. IPTG bind to the LAC-I repressor proteins, which then detach from the DNA. This allows RNA polymerase to transcribe the gene before IPTG is added. The LAC-I repressor prevents expression of the clone gene. Now cloning expression, and one thing I added, the subtle vectors. The cloning vectors is a small piece of DNA that can be stably maintained in an organism and into which a foreign DNA fragment can be inserted for cloning purposes. And expression vectors are associated with the actual expression of the gene into mRNA and protein in the target organism. And a subtle vector is a vector, usually a plasmid, constructed so that it can propagate in two different host species. Therefore, DNA inserted into a subtle vector can be tested or manipulated in two different cell types. A subtle vector is plasmid engineered in a way so that it can propagate in two different hosts, which is bacteria and yeast for this vector. And along with this, one other thing is TA cloning. When we don't have restriction site to isolate our desired gene to combine with vectors, we use this method for cloning purposes. TA cloning is a sub-cloning technique that avoid the use of restriction enzymes and is easier and quicker than traditional subcloning. The technique relies on ability of adenine and thymine on different DNA fragments to hybridize and in the presence of ligase become ligate together. Here, vectors having protruded T base and the insert that containing desired gene is having A base pair at 3 prime. Now these join and A pair with T and forms TA cloning vectors. This is example of that. By using this, we can clone the desired gene which are not having any specific or available restrictions sites. Now I inviting to conclude today's session Professor Dr. Archana Tiwari ma'am, Director, School of Biotechnology, RGPB Bhopal. Over to you, ma'am.
Thank you, Dr. for wonderful presentation. Very informative. Now the time, just to wind up our session, that is what we have discussed today. I'm extremely sorry because the sessions are too late, only because of the informations which are already present in that session. We have to deliver, we have to discuss that with you. Otherwise, if we will give just in superficial, so that we cannot justice with that topic. I'm extremely sorry because the session is too late. only because of the information which we have to just share with you. Now, only quick 2 minutes for just review what we have discussed today within the first two sessions. Cloning of DNA fragments, that is with the help of molecular or DNA cloning. Basics, the purpose of this workshop is we have to focus on the basics of the subject, because whenever we are dealing with all these, we are just ignoring some of the basics. That's why our main motto is, whenever we are going to start our work, we have to be very cautious about our basics when we are designing our work or going through any of the ways in the field of biotechnology. So, in the first session, we have discussed the three types of cloning: molecular cloning, reproductive cloning, and therapeutic cloning. While in the second session, that is cloning vectors, expression vectors, and other vectors, as Dr. Mrityunjay has just some of that. I know today the time is too long, but we are leaving here and meeting tomorrow at the same time on the same platform. With this, you are joining us tomorrow also to complete this workshop. Till then, goodbye, take care, and enjoy yourself, and take as much as vitamin P you can take, right? And always I'm saying P stands for positivity. With all these, thank you so much and namaskar. Now, we are going to end the meeting today.